Okay, so thank you very much for attending this discussion, and also thank you very much for Apical Science to organize this webinar. So today's topic will be about anti-contamination. Basically, when you work with cell culture, this is one of the major things that you have to keep in mind. So first, you have to know your risk. Where are the contamination? And then how to protect yourselves. So before we go into the discussion, just to give you a very brief introduction about InvivoGen. So we are established in 1977 in France, Toulouse. And then later we set up a subsidiary in San Diego, US. And later another headquarters in Asia, Hong Kong Science Park. So since 1977, we have been focusing on developing antibiotics to help our users protecting their cells. And so contamination. So this is the question that we commonly see from ResearchGate. So this is one of the questions I adopted from ResearchGate. So as a source of contamination by cell culture. So a lot of the time when you go into your laboratory, when you take a look at your cells under microscope or just by observation, unfortunately, you may be able to see some strange thing. For example, on the left, this is your common cell culture. So you have the cells at the background, pretty much 100% confluent. But then you also see some other thing on top of it, which doesn't look very like your cells. Same thing for this naked eye inspection. Sometimes in the cell culture medium, you may be able to see some cloudy stuff in your culture medium. So these are signs of contamination. And this is probably the worst thing that can happen to your cell culture. So where do they come from? It can come from a lot of different places. For example, your film hood, the ventilation unit, of course, the operator. On your body, you will have a lot of microbes inside your body, inside your mouth, that may be spread to the cell culture. Pipettes, tips, syringes, if they are not properly sterilized, they are also sources of contamination. And of course, culture media, reagents, and cells are some other source. But basically, with now, many more people are paying attention to the aseptic technique to have a better control or management of their equipments and also the culture media. It is quite unlikely that the source can come from this hardware. Cell culture media, or for example, FBS, was a major source of Microplasma contamination a long time ago, but not now, because the standard has been upgraded and more people are aware of this. So the manufacturer, they have already paid special attention to keep their reagents clean. So this was a major issue in the past, but not now. Same thing for pipettes and tips. You always autoclave or use the already packed, sterilized pipette tip box when you work with cell culture. So these two are not likely. And if we look at some feedback from some of the experts, for example, Hans Drexler, the head of DSMZ. So DSMZ is a cell bank in Germany, pretty much the equivalent of ATCC as in America. So this is one of the best and biggest cell bank in the world. So DSMC head Hans Drexler mentioned this before. Cell cultures, they don't spread mycoplasma or bacteria or fungal or other contaminants. It's actually the people who are handling the cell cultures. So from him, the major source is the operator. So we have to wear gloves, we have to wear protective clothing, and most importantly, don't talk when you're doing cell culture. Your mouth, there are lots of mycoplasma and maybe some bacteria, and those aerosols, when you talk, can be easily spread to the cell culture. So people are one of the major factors, because others we can control. Tips, cell culture media, these hardwares we can control, we can clean, but people are the main source. So take-home message, do your protection when doing cell culture and don't talk. So this is a source of contamination, but a lot of the times we may not be able to 100% prevent. So we have to know the sign and the types of contamination that we are encountering. Major types of contaminants when doing cell culture can be grouped into 5. Mycoplasma, bacteria, and yeast are the most common found in cell culture. And sometimes you will have eukaryotic cell contamination, which means when you are culturing different cells, you are not careful enough so that you cause the different cell lines to mix together. You're culturing 293 cells, and then you may be able to see some other patches of the cells that look differently from a 293, but they're growing happily. No sign of contamination, no yellow culture medium, no turbidity. But if you can see 2 types of cells growing in your culture plate, then it probably means that you contaminate or cross-contaminate the 2 cell lines that you're working with. But this is not common, and also virus can be a source of contamination, but again, it's not common because it's very difficult to infect the cells with a virus from outside, out of nowhere. So the most common contaminants that we are daily facing in the laboratory contains bacteria, yeast, and mycoplasma. Sign of contamination. So it's very easy. If you have some experience with cell culture, you may already know that bacteria and yeast contamination will lead to yellow medium, or even you can see cloudy medium. So that means you have contamination. So from this example, you can also see the different meanings of different look of the medium. So for example, if your medium looks like this, you can leave it alone. If it looks like this, then you have to prepare for medium change. And if it looks like this, then you have to check for contamination. So this is the sign of contamination. And so because in InvivoGen, we are also doing research and development, and we work with a lot of cell lines. So apart from antibiotics, one of our major focus is innate immunity. And to study immunity pathway, we work with lots of immune cell lines. So in-house, we're also paying a lot of attention to contamination. So with this, we have developed some tools to help doing cell culture. First one is for prevention. Second one is for detection. And then last one is for elimination. So I go through it one by one. So for prevention, as mentioned before, bacteria, yeast, and mycoplasma are the major source of contamination. So to prevent it from happening, we have developed several tools that can prevent all of these contamination. So it's like one for all reagent. So we have two of these one for all reagent, Normosine and Plasmozine. So Normosine can be used as a daily protection reagent. Some of them may have heard of penicillin streptomycin. So PS is also one of the major addition to your cell culture medium to prevent contamination. But the little disadvantage or limitation for pen-strep is that it can only prevent against bacteria, but not yeast, but not mycoplasma. So if you use pen-strep, then you may have to find another method to prevent yeast and mycoplasma contamination. You may need to add some more reagent, which sometimes you're not sure whether they're compatible with each other. So to solve this, we prepare a one-for-all solution, Normosine to prevent everything, and also Plasmozine to prevent everything. Now let's take a look at Plasmozine. So we have two one-for-all reagents. So what are the differences? So Normosine is for common cell culture. While we notice that some researchers working with a special culture, they may need special attention. So that's why we developed another product called Plasmozine. So again, it's active against bacteria, fungi, and mycoplasma. But the one thing that makes it stand out is that it is designed for primary cell cultures. Not your common HeLa or 293 cells. Those are tougher cells. So we can basically use stronger or slightly more toxic antibiotics to achieve the prevention or elimination need. But for primary cell cultures, especially for stem cells, if you add something wrong, they may differentiate, and then you can no longer have the stem cell that you like. So for these cells, it needs special attention. And so we developed Plasmozine for primary stem cells, primary cell cultures, including stem cells or 3D cultures. So this is an example from the publication. This is the patient-derived organoid generation. And here they are comparing the treatment they have in mind after getting the samples to see how they affect the samples or how well they can protect the samples from contamination. So after getting the samples, you may do no wash, and then you can see here the culture medium become very cloudy, and you can see different patches, which is most likely to be bacteria or fungi contamination. If you wash with PBS, it doesn't really help much. You can still see the patches. You can still see the cloudy thing in the cell culture. PBS with pen-strep, still, it's not very nice. So this is showing that another limitation for pen-strep. They may be able to protect against some type of contamination, but not everything. So that's why even if you add pen-strep, if it is something that they cannot cover, you will still see the contamination. Now this is with Plasmozine. After adding Plasmozine, it's pretty much clean, at least for naked eyes. It is much cleaner than, of course, no wash, and even much cleaner than pen-strep. So again, we can tell that Plasmozine is superior in the protective function. If you mix Plasmozine and pen-strep, they are compatible and they keep the cell culture clean. But if we compare to this sample, adding only pen-strep, we can basically conclude that the effect of protection is not likely to come from pen-strep. So first, when you generate organoid samples, this publication shows that Plasmozine can help you protect the sample from contamination. So how about the bottom part? The bottom part was that the author, they did an extra check to see how safe is pen strep and Plasmozine. Most of the time when we work with antibiotics to protect contamination, protection is one thing. Safety is another major concern. If it's toxic, then there will be a limitation of where we can use the reagent. So this is protection on the top, and then the bottom is the safety. We're looking at live cells percentage, PBS as control. And so this is PBS, and then you can see pen strep. At the first look, you can already see pen strep is usually lower than the PBS. So for example, compared to this, compared to this, compared to this, the pen strep compared sample is usually has less live cells compared to PBS. So it suggests that pen strep in this setup, in this organoid culture case, they showed toxicity. But for Plasmozine compared to PBS, there's no major difference. One or two samples, you may see a major difference, but in general, if you look at the statistic significance, the difference is not significant. Plasmozine doesn't cause major toxicity to the organized cell culture. So from this, we can tell that if you're working with primary cell cultures like stem cell or 3D cultures, Plasmozine can be used as a high efficiency and low toxicity reagent to protect your sample. So that was about prevention. Now let's take a look at detection. So for detection, we will pick out Microplasma as the special one. Why? For bacteria and yeast contamination, you don't really need a specialized detection method. You can see it by naked eyes or simply by light microscope. The medium will turn yellow or turn cloudy. You can see it right away once you open the door of your incubator. You don't even need to do special tests. If you want to confirm, you can always look at it under light microscope. And bacteria and yeast can be picked up by light microscope. So for these two, you don't worry. You don't really need detection method. Microplasma is the special one. Why? Because it's so small, 0.1 to 0.3 micron. It cannot be picked up by naked eyes or light microscope. So it is smaller than the detection limit of the light microscope. So you can have a very severe contamination of Microplasma, but it looks just fine under a microscope or by naked eyes. That is the major issue for Microplasma. So that's why compared to bacteria and yeast, we need some specialized method to detect it. And then we can set up the solution to eliminate the contamination. Another major headache for Microplasma, again, is because of its small size. Commonly used filters in cell culture are 0.22 micron. We filter our medium, we filter our drugs using 0.22 microns. Microplasma having a size of 0.1 to 0.3 microns, they can pass through the filters. Filters have no effect to them whatsoever. So it cannot be detected easily and it cannot be prevented easily. So that's why we need to set up special ways to detect and eliminate Microplasma. We have two ways. The first way of Microplasma detection is by a reporter cell line. We know Microplasma is a microbe for the cell. It's a invasive thing. It's an outsider. So actually in our immune system, we already have a built-in system to sense mycoplasma and then to clear mycoplasma, because mycoplasma also infect animal and human. So in our body, we use a receptor called TRR2. Once it senses mycoplasma, it will activate the NF-κB inflammatory signal. So adopt this pathway, build a stable reporter cell line by connecting the NF-κB to a alkaline phosphatase reporter gene. When this reporter cell line senses mycoplasma, this pathway will be activated, producing the alkaline phosphatase. When the alkaline phosphatase is produced, we can add the substrate, and then it will give us blue color. So blue color means mycoplasma positive. Pink means negative. And because it's a colorimetric readout, it can also be made quantitative if necessary. So the good thing about this system is because it is based on our immune system. This system has been optimized for millions of years. So it can detect all mycoplasma and ecoliplasma species known to infect cell cultures. 100%, not 90, not 95. It can detect all mycoplasma and ecoliplasma species. It is easily scalable. You can do a 96-well plate. If you want more, you can upgrade it. So let's say if your laboratory is working with a lot of different cell lines, if you are the lab manager or if you are helping to maintain a cell bank, you have lots of cell lines to keep track of, then this can be a very good method, because even if you have 200, 500 cells, you can easily scale up to do your test. And again, because it's based on the color, the more blue, it means the more mycoplasma you have in the sample. So it's quantitative if it is needed. The protocol is also very simple. You collect your sample that you want to test, you heat it for 50 minutes, and then you add the samples to the detection cells, mycoplasma sensor cells, which is this one. After that, you incubate, and then you add the substrate. And after adding the substrate, the mycoplasma positive one will turn blue, and then you can detect it by the absorbance. So this is one way to do it. It is suitable for large-scale screening, but it is not very friendly for small scale. So for a more flexible and small scale test, we develop a completely different method. This is the solution we provide for small scale and flexible screening. This is pretty much like the quick test for COVID-19, but the basic is quite different. So it is not detecting the antigen. It's actually still PCR based. And this is a protocol. You collect the sample. There's no special treatment. You simply collect the cell culture supernatant, and then you add in the reaction buffer, mix them together, incubate 65 degrees for 40 minutes. And this is the PCR step, 65 degrees, 40 minutes. After this, you simply put the reaction product into this hole, and then it will give you the result. The readout is similar to the COVID-19 test. 2 stripes means positive, 1 stripe means negative. So after all, it is a PCR base, so it can keep this sensitivity although we make it much simpler, we don't sacrifice the sensitivity. But the beauty of this is that for traditional PCR method, you may need to extract the DNA from your cell culture sample before you do the PCR. And then after PCR, you have to run the gel and then visualize it. For this test, we simplify before and after. Before PCR, no sample pre-treatment. You simply take the cell culture supernatant and you're ready to go. After the PCR, no gel running. This replaces the gel running part. So it's much easier to use, and you don't even need a special equipment because the only special thing about this protocol is the 65° 40 minutes. You can use a water bath, heat block, or if you have the PCR machine, you can always stick back to the PCR machine. This is the only special equipment you need for this kit. And it's quick, 1 hour and 15 minutes hands-on. So most of the time, you're waiting for this 40 minutes, while the before and after, they are super simple. It's simple, but as mentioned before, it keeps the sensitivity because it's PCR based. So this is the new method that we came up with. And so we contacted another cell bank and they're interested. So the cell bank that we are now working with is Jcrb cell bank. So at the very beginning, I mentioned the DSMZ cell bank from Germany. And this Jcrb cell bank is in Japan, which is another very famous cell bank in the world, equivalent to ATCC in the States. So Jcrb cell bank help us do some tests on the microplasma detection method, and then they presented the result in the annual meeting of Japanese Tissue Culture Association. So this is the original poster in Japanese, but I can walk you through what they found. So to compare the sensitivity or the performance of different methods, they came up with a set of samples. The first one is the concentrated Microplasma standard. The next one is the dilution to the enzymatic assay detection. So they try to use the enzymatic assay as a standard, and then so that they can compare other methods where they are higher or lower than the enzymatic assay. After that, if we all pass this detection limit test, they will further dilute into 10-fold, 100-fold, and 1,000-fold for further analysis until no one can detect anymore. So this is the setup of the samples. And they tested several methods. The first one is the enzymatic assay. And then the second one is the microstrip from us. The third and fourth one are the more traditional method, traditional nested PCR and Hosch staining. So Hosch staining is actually staining the DNA content. So you can see here, you can see some network-like structure next to the nucleus. So these are the microplasma that can be stained by the Hosch staining. All right, so first they look at the concentrated standard. For enzymatic assay, it's positive. Microstrip, positive. That's the PCR positive, and Hosch staining is also positive. So of course, it's very concentrated sample, so we'll be very surprised if one of these methods could not detect it. Now here comes the real deal. Enzymatic assay limit as the standard. So of course, it can detect by the enzymatic assay. Microstrip also 2 bands, positive. Nested PCR, positive. Hosch staining, positive. All pass, so we move to the next step, 10-fold dilution. After 10-fold dilution, the enzymatic assay could no longer detect the microplasma, although there was still microplasma inside, because we know as we prepare the dilution sample. Microstrip, 2 bands, still positive. Nested PCR, still good. Hot staining, still good. So how about another tenfold? After another tenfold dilution, of course, enzymatic assay, they failed. Microstrip, we could no longer detect it. Hot staining, negative. PCR, uncertain. So they have done twice, and then 1 positive and 1 negative. So we're not sure. Although we know there were microplasma in the sample, but if it is a blind test, 1 positive, 1 negative, we cannot be sure. We may have to do an extra test. So this is uncertain. And of course, they also did 1,000 and 10,000 dilution, but basically it's the same thing, or like everyone cannot detect. So from Jcrb cell bank, they concluded that microstrip, our novel microplasma detection kit, has similar sensitivity compared to traditional PCR, which a lot of labs are using, and also hot staining. They have similar sensitivity. Although it's much easier, we did not sacrifice the sensitivity. And this is the general result. Concentrated standard, all positive. Enzymatic assay limit, all positive. If we do tenfold dilution, this one failed, while others are positive. 100 dilution, all fail. PCR, not sure. So basically, the reliable sensitivity, they're in the same range. So this is the major feedback they provided to us, and it proved that our concept is correct. We can make this PCR testing method simpler, but keeping the sensitivity. So this is the beauty of this novel detection kit. So we talked about prevention and detection. So how about elimination? Unfortunately, once we have the contamination, to be honest, the best advice will be to throw it away, because it can prevent other cell culture or the lab environment in general to getting contaminated, or to avoid the spreading of the contamination. But there are times that the cell line is so valuable, maybe a stable cell line, maybe a primary cell line or cell line from a patient that we just cannot discard. So for this, we will have to do the elimination or treatment process. So again, let's take a look at Microplasma first, because this is the most problematic one. For bacteria and yeast, it can be spotted easily. So even if you have to treat it, most of the time you can treat it very quickly in time. Well, for Microplasma, because it cannot be detected easily, the contamination can last for very long and so concentrated that sometimes it cannot be treated easily. So for Microplasma elimination, we have this Plasmozine, which is the best seller of all our products. So this is well trusted by our customers and well cited in the publication. So it's targeting Microplasma, effective and safe. And this is the general treatment using this drug. You simply add the drug to cell culture, 4 packages over 2 weeks, and then you can perform another test. Most of the time should be clean, but if not, then you can continue another 1 week, and then you can detect the Microplasma again. So this is the general way for us to treat Microplasma. And again, similar story as mentioned before, treatment is one thing, safety is another. So there was one group, a third party, not by us. They published in PLOS One thoroughly testing of Plasmozine in human embryonic stem cells. So here they're comparing two sets of cells. The first one is the control, which they call H5. And then the other group is the H5AA4, which is the contaminated cells and treated. So they're comparing different aspects between the control and the contaminated and treated cells to show that this treatment, this drug, is safe. So first, by PCR, they detected the Microplasma contamination. So you can see this is the infected, very, very strong band. This is a control, so you can compare. So first, they finished the treatment 14 days, and you can see this is after 14 days of treatment. The band is gone. It's the same as the H5 negative sample, clean. The next thing they would like to confirm is that whether this is really elimination or only partial suppression. Because if it's partial suppression, you can also see this. But then once you remove the drug, it will pop back immediately. So it doesn't solve the core problem. So they did the test 6 months after the Plasmozine treatment, and still they can see it's completely clean. So it shows that this drug can really 100% eliminate the contamination, not partially suppression. There's no relapse. This is only a treatment part, but we want to know the safety. So they compare the H5 and H4 in different aspects. First one, by light microscope, they could not see any major morphologic change. Second, since this is human embryonic stem cells, they look at the stem cell markers in both the protein and the mRNA, comparing H5 and the H4. There's no major changes in the protein, and there's no major changes in the mRNA level, H5, H4. They also look at the chromosomes, and they could not identify any abnormal observation. Next, they put the test to a next step. Since it is stem cells, they want to see after treatment whether it will affect the stem cell differentiation. So they took the two cells, H5 and H4, and then they differentiate the cells, and then they measure the gene throughout the 14 days course, and there's no major difference. And after that, they really look at whether the two cells can differentiate into ectoderm and endoderm to really look at the pluripotency in real situation. So H5, H4 can both be differentiated into ectoderm and endoderm. So it shows that Plasmozine is completely effective and at the same time very safe. So in this case, they are working with human embryonic stem cells, which is super sensitive. So if it's safe in stem cells, in general, in theory, it can be also safe in a lot of daily cultures. HeLa 293s, they are known to be the, let's say, tough cells. So there should be no problem using this drug on these two cells. So that was about Microplasma. Now, this is another interesting story, which concerns another type of contamination elimination. So that was Microplasma, but then this is about bacteria. So this scientific reports, they had a very imaginative approach in collecting cells from animals. So they try to study the non-human primates like chimpanzee, like monkeys, and they want to get the urinary cell samples from chimpanzees, monkeys, or gorillas. Due to practical and ethical reasons, it is quite hard to find an easy way to collect the urine samples from these non-human primates. What they originally planned was to collect the urinary cells to make iPSC, but then they couldn't find a proper way. So they had an imaginative idea that they asked the zookeepers to collect the urine samples from the floor, from the zoo floor, which you can imagine how dirty it can be. So at first, when they are trying to capture the samples from the zoo floor, they failed due to the growth of bacteria. And so from the methods and materials, you can tell that the solution that they found was normal cure from us. Isolation and culture of urinary cells only became possible upon the addition of normal cure. So this normal cure thing is the antimicrobial reagent that specifically target bacteria. So it does not only prevent, but it kills. So it can be used even after the contamination. And this is exactly what they are experiencing. They already know they have bacteria contamination in the samples. So they add the normal cure to make it clean so that they can work on the samples collected from zoo floor. And the cells can also be derived into iPSC, meaning that the normal cure is very safe and it doesn't affect later experimental procedure. So this is a very interesting use of the product. Normally, we only expect it to work in a laboratory situation. For example, you have a small-scale contamination of bacteria, then it may work. But then this scientific reports actually show that it can work in much worse situation, much worse than we expect when we develop this product. So this is very nice and also shows the power of normal cure. So this is one thing, but of course, not many of you will work in this setup. But there's another quite common example that may suit you. Not only for mammalian cell culture, but let's say if you are working with fungi, you want to collect your special fungi sample from soil, from the ground. A lot of the time when you collect samples from the ground, from the soil, they will have bacterial contamination as well. In this case, this will be a very good option for you to clean the bacteria contamination so that you can get your special fungi sample from the floor or from the soil. So apart from mammalian cell, this is another possible use for this powerful cleaning reagent. So today I have shown you that the source and the risk of contamination is basically everywhere, but especially from the operator. Sometimes it cannot be completely prevented by hardware. It cannot be completely prevented by wearing gloves or by using clean reagents. So to protect ourselves, we may need to use some special prevention measures. For example, Normozine and Plasmozine, which can cover all the common contamination source. For detection, there's no need for bacteria and yeast because you can see it right away. But for Microplasma, again, you need some special method for detection. So here I've shown you the special new microstrip kit that we developed and also verified by the Japan Jcrb cell bank. For elimination, I've shown you the use of Plasmozine to treat the human embryonic stem cells, high efficient and high safety. And also normal cure with a very interesting and special story about the samples collected from zoo floor. So if you need any more information, you can go to our website, and we also have a more comprehensive cell culture contamination, which covers everything that I mentioned today, and also some additional information that I have no time to go through today. So this can be downloaded from our website, and you may go to here for more information. And with this, I'll end today's presentation.